get going.
So, first of all, thank you so much for having me today. My name is Kimberly Munn. My background is I'm an oncology nurse, and I currently work for Bayer Pharmaceuticals, but this is not on behalf of Bayer, and nothing is endorsed or represented by them. It is by me solely as an oncology nurse. And today we're going to talk a little bit about genomics and genetics, and that's just kind of becoming the common language when we talk about cancer. So, I mean, really, we're starting to think about it even beyond cancer as well, but definitely one of the bigger areas in which I think a lot of things are changing. So I have a pretty complicated slide that I'm showing you. And why I want to show this to you is that this is a lot of times what drives what we do when we're thinking about treatment of cancer or what we're doing in the research of cancer. So like, we're going to go back to biology days and remember when we first had to learn how to make a cell, and that a cell had a mitochondria and it had a nucleus and it had DNA in it. And there were all these little things and proteins that mattered in terms of how it behaved, right? So we had to learn how a normal cell behaved first. What's interesting is cancer are your normal cells that kind of went bad. They kind of mutated. They altered in some way. And that is what the hallmarks of cancer is. So the hallmarks of cancer is the terminology we use when we talk about how the biology of cancer behaves. So we know it acts different than a normal cell, which is why we have to go after it in a different way than we would with a normal cell. So I'm going to just give you one or two examples on this list to help you understand that. So one of the things that, let's see, I don't know if you can see my cursor or not. In the lower corner where it's kind of red and it has a vein and it says inducing or accessing vasculature. Okay. So that's the ability for cancer to push out some like signals to make its own blood supply. That's a special thing that cancer cells do. And so there are certain cancer cells that do it more likely than others. And some of those cancers are brain cancers. Another cancer that does that a lot is multiple myeloma. There are certain cancers that they use that mechanism in a way overly than a normal cell would. And so we have drugs that stop that. And one of those drugs would be something like bevacizumab or Avastin. So that is an anti-VEGF drug. And what that does is it stops the cascade of VEGF, and VEGF is the vasculature epithelial growth factor. And so that's just one area in which we have one small part in which we're trying to utilize that to help stop cancers from growing. But there's all these other ones as well, right? There's 14 different dimensions here. So evading, your immune system is one, like there's so many here. So that's why I wanted to give you this, because I think that helps you understand why we have why we do what we do, right? Why do we have to get so much information? Why do we have to look at your cell so minutely all the way down to its DNA and RNA? Because that's going to give us the clues on what it's doing to keep evading the things from being, that's how we have to treat it, to get rid of it. I am going to go to a different slide, because now I'm going to take you on a small journey. And this little small journey is kind of the genomics, what has happened in the world of genomics in oncology.
I think that's the same slide, though. I don't see it.
Did you not? You know what? I might have to do this. I might have to do a new share. Sometimes I have to. Do my screen. How about now?
Yes.
Do you guys?
Yes.
Thank you. That was so helpful, because down in the bottom, it looked like I had it right. Thank you. So we're going to just go quickly through this little milestone, and it's going to kind of help you kind of shape the it's actually been around for quite a while, right? So we started the discovery of DNA, the structure, basic structure of it in 1953. By the 1960s, we learned about something called the Philadelphia chromosome. That's something that was discovered that's unique to patients that have CML or chronic myeloid leukemia. The other name that we use for that is BCR-ABL. That's the actual gene that we talk about when we're talking about BCR-ABL. This is the special chromosome that they realized was unique. And when they saw that, they were more likely to realize that they had this type of cancer and was a potential target. We go to the 1970s. We have a National Cancer Act that's signed. Tamoxifen is actually the very first targeted therapy that's ever approved. And tamoxifen targets estrogen. So interesting enough, hormones are definitely one of those things. And two types of cancers really push out for hormones, right? Prostate cancer uses a lot of hormones, and breast cancer, and some of the gynecologicals. So if you can shut down the hormone that cancer is using to feed itself, then it's one of the ways to treat your cancer. Then there's something called the TP53 gene. TP53 gene is interesting because that is the most common altered gene in all of our cancers. We can't target it yet, but if that ever happens where we can actually treat the alteration of TP53, that will be huge. We go to the 80s, HER2, which is breast, it's a human epithelial receptor that gene HER2 was discovered, and that drastically changed how they treat breast cancer because they are able to make Herceptin. And now we have genome programs where we're actually doing our first meeting from the time we figured out DNA. Just about 40 years later, we're having our first ethical, legal, and social implications working meeting for the Human Genome Project. We go to I missed the 1990s. This is where we start to learn about germline or genetic factors that you're born with that potentially put you at risk for cancer. BRCA1, BRCA2, Rituxan or rituximab is our first monoclonal antibody that's approved, and that's something that tacks onto something called CD20. Then we have trastuzumab, which is for that HER2 target that I talked about earlier. We go to the 2000s and it just accelerates, right? So now we're like kind of moving along, and then we're going to like explode like a rocket now. Literally 2000s beyond, things are happening super fast. Now we have technology. Now we have all this stuff that we can do that we never were able to before. And so imatinib is actually the very first drug that is made for that BCR-ABL, that Philadelphia chromosome that was found back in the 60s. And it's wonderful. It's one of those drugs that drastically changed the game for people who had that type of leukemia. It was I remember, because I'm a nurse from the 2000s. So I remember giving that drug and seeing patients who used to die within a year living for like 15 years. It was beautiful, right? And so we're like, man, we need more things like that. Then you have your Human Genome Project, and then you have your Cancer Institute does a study with tamoxifen and raloxifene for chemo prevention. So it's a whole way of how can we have less chemo and less toxic therapies if all we really need is just that targeted therapy. And like I said, here's where it just goes bananas. We get ipilimumab, which is one of the first immunotherapies that comes out. You have your Cancer Genome Atlas project that happens, Pembro, which is the main immunotherapy everyone thinks of, which is Keytruda. And it's approved for multiple tumor types, which is also really exciting because before it used to be very specific to what type of cancer you had. That one's approved for many types of cancers. That's a big deal. Something called MATCH, which I actually was a research nurse and did MATCH trials. And so that was really cool, where they take pieces of your tissue, they look at your DNA, and they offered different drugs that may not have been approved in your disease type, but because your cancer showed you might have efficacy for that, they could give it to you. And then we have the Moonshot Initiative. Then there's pediatrics that comes on. I mean, you name it, just lots and lots of stuff. And you know, just recently, the Moonshot just got reignited. I'm going to stop there and just make sure there aren't any questions, or can you see that I switched to a different site? Okay.
Yes.
Any questions, comments, concerns, or genius ideas? All right, I'll keep going then, and we'll kind of talk a little bit more about language, right? Because essentially what I wanted to talk about, and this is something that we can go over with our patients, and so it's a really great tool that Oncology Nursing Society has, is kind of like when we talk about genomics, we're really talking about usually something called next generation sequencing, okay? And so what basically happens is every cancer has its own unique genetic makeup. And we are doing biomarker testing that really helps us collect that information about the cancer so we can personalize the treatment that fits best for what your cancer's biology is showing as how it works, right? As how it behaves. All right. And so sometimes you'll hear other names for biomarker testing as tumor genetic testing, tumor profiling, molecular testing, genomic profiling. Those are all kind of the same basket. They're pretty much talking about the same thing. And so what that biomarker does, what we're doing is we're testing to check for the changes in your gene, or proteins or other molecules that kind of help provide how that tumor's makeup is. So remember that big complicated slide in the beginning? We're looking for all of those things. We're trying to figure out, okay, is it hormones that it feeds on? Does it like to send off information to try and make blood supply? Is it telling your own immune system to shut down because your own immune system actually knows cancer shouldn't be there and is constantly usually getting rid of it? So why did it stay? Because it's making it go to sleep, right? All these things are what we're looking at. So one of the most commonly used, like I said, was next generation sequencing, and that kind of allows to have many biomarkers tested at the same time. So we're talking about, 500, 600 gene types. Like it's a pretty significant amount of information that you're getting. It's also usually the most comprehensive, the fastest, and most cost effective, even though there's lots of different ways that you can get testing done. I want to say, I think this one goes over some of those. So I won't go too far into that until we get there. So, the main reason why we like to do it before you get your treatment, right? And so that would either be getting a biopsy, maybe some blood samples, probably both, is because it could actually change the treatment you're going to get pretty drastically. And we wouldn't want to give you something that's not effective for you, just to start something. It's actually, there's studies that have been done, and especially within like lung cancer is a good example, because lung cancer actually probably has the most biomarkers right now that it utilizes to determine treatment. They're pretty advanced in this area. And there's evidence to show that you have a worse outcome if you give certain treatments before knowing the biomarkers, if you don't treat the biomarkers that you should treat first. It's actually worse. So, this is like an important thing to kind of learn, because I think when you first find out you have cancer, the first thing you think is, get it out of me. I don't want it, right? And so it's really hard to hear that, to be like, oh, man, it's actually important to get the test done, to wait for the results and let that guide you on what you should do. Of course, if you're having certain symptoms, there's always the discussion of starting certain things if you're in a situation what we call symptomatic. All right. So then, we select this biomarker. We're going to perform that test. The test usually takes about 3 weeks. It depends on which one you send, but tissue, which is what we call the gold standard, really takes about 3 weeks to get back, 3 to 4 weeks. And then that's kind of really, we analyze those results, and on that, it gives a whole bunch of information. All right. And like I said, why do we use it? We want to use it to understand what therapies we should give you. We sometimes can know what therapies won't work on you because of a type of biomarker that you have. And then even more importantly, and something that I'm always going to be a big advocate for, is if you're eligible for a clinical trial. We are now at the time where I would say a clinical trial, if you are eligible for a clinical trial, you should do it first. That's everything that's kind of currently coming out in terms of being exposed to clinical trials will likely be one of the best treatments that you can get. So don't wait for a clinical trial to the end. You should be thinking about it almost at the beginning if you can. So let's talk about what's this next generation sequencing, right? What do you need? So like I said, it can be on blood, it can be on body fluid, or it can be on the actual tumor tissue. Okay. And then
one question.
Oh, sure.
If you're waiting 3 weeks approximately for the results, and then you're in a clinical trial, to me, a clinical trial means that there's nothing concrete. It means that you have more time when you have nothing definitive to go on.
So you know what? That's a fair point. That's a fair point. The interesting thing is, by the time these treatments get to human trial, it would be unethical for us to ever provide something that isn't as effective as or better than the standard of care.
Oh, okay.
So that's the I'm so glad you said that, because right, that is one of those kind of thoughts, and that's one of those ethical, moral things that we've set up to make sure that you would never get something that's less than the standard of care that you would get. You need to have at least standard of care, if not better. So that is that's a great point. Thank you for asking that. But again, always important. No one ever, you can always say you want to go and do a clinical trial and come back out. You can say, like, I'm not this time, I want to do it for next time if possible, because there's lots of treatments out there. So always make sure to have that conversation with your oncologist for sure. So your next generation sequencing, a lab test is going to be done on blood, fluid, tumor. It ranges anywhere from single gene testing all the way to what we call whole genome, which is where instead of just taking like strands of your DNA, they're literally doing all of your DNA. They also do RNA, which is even a whole nother set of information that helps actually tell you what the true building blocks are. Because we know DNA has a lot of information, but we have DNA that doesn't do anything, right? We have DNA that never actually shows up phenotypically what it should, what it would make. It's just the potential of what it can make. So RNA is a better example of what's actually being created within the protein. And really, this kind of varies. This is probably the one where like the testing is kind of what your doctor feels most comfortable with, or what's available to them, or what your insurance pays for. So that one's another one where you can do a little dabble in, you can, call your insurance company to see what they cover, what they prefer. You can talk to your oncologist about it. Some places are, offer zero copay. So, there's lots of potential there to have a bigger discussion about which ones they decide. But that's more kind of really usually determined by your physician. The actual testing, once they take the tissue, the blood, or your body fluid, they'll take it to a lab, and that's where it's processed and analyzed, and that's where it can take up to that 3 weeks. And so, why is it important? We talked about this. We know that there's better survival rates when a therapy that's specialized to that biomarker that you have, than just a one-size-fits-all chemotherapy. There tends to be still side effects, but different side effects, and tend to be fewer side effects as well than chemotherapy. Because chemotherapy, we know, is wonderful in the sense that it's a very effective treatment. It's just that it destroys all the cells that are rapidly dividing. And that also means your good, healthy cells. And that's why, you know, your immune system falls and your blood level, you know, you get anemia and you have a higher potential for, you know, vomiting, and you lose your hair, like all of those things, because that's what that chemotherapy destroys as well, unfortunately. Targeted therapies tend to do a little bit less of that collateral damage. So once that test is done, your results go to your provider, and then they, you know, can share and talk about that report with you. That report is pretty high level, so there's going to be a lot of words on there that can be confusing, and that was going to be my last spot if we have time, but I already know we're a little over, so I can try and wrap this up a little bit. Okay. Actually, this might be a good time, because this is what a lot of things about what we would talk about. Okay. So there's two things we think about when we are looking at the DNA of somebody, the tumor or the person. There's something called germline or inherited, and that is what your cells and your DNA have already all the time and what you're born with. And then there's something called somatic, and somatic or acquired, that is what happened to your DNA after some type of exposure over a lifetime. All right. And that's really what we mean when we look at your tumor. We look at germline because there might be things that we need to look for that maybe you were born with. But the most stuff that we're looking at is called somatic or acquired. So what were the alterations or the variants that developed that your tumor is showing us as being something that's either benign, harmless, pathogenic, harmful, or we don't really know. And those are called variant of uncertain significance, or VUS. We don't really have enough information that's whether it's harmful or harmless, but I can tell you in that little spot of VUS, variant of uncertain significance, about 2% of the things that we do end up finding, what they are, end up actually being harmful. The majority end up being harmless. So the good news is VUS is more likely benign or harmless than it is harmful. So that's why we just kind of look at it, and we're waiting for more information in a couple of years to see if, you know, if it ends up being something that matters. So I like this because I like the little kind of recipe of what each of these mean when we're looking at different things. So, you know, benign is like a harmless change that does not lead to cancer. So if we're looking at your report and it says benign, it's probably like a blueberry cake that calls for a stick of butter. Unfortunately, we only have margarine. So we substitute the margarine for the butter and we bake the cake, and it tastes delicious and it's getting great reviews from the family. And so that's like a benign variant. Okay. There was a change in the recipe, but it didn't affect the taste or the consistency of the cake. So it was a harmless change. Okay. Now we have next, like I said, most likely benign, but we're not really sure, right? So we have a variant of uncertain significance, and that's like a chocolate cake that caused for a stick of butter. We only have margarine, which we substitute for the butter. We serve the cake, and some family members like it. Some say they don't like it. They think it tasted a little weird or wasn't as very good. And that it's kind of just like unknown, right? The meaning of the change is not clear. In this example, this is not agreement that the impact of the ingredient situation in the recipe, right? So we need more information, and maybe some people just have really, really good taste buds. I don't know. And then pathogenic, meaning harmful, is we have a lemon pound cake that calls for a stick of butter. The recipe emphasizes avoiding any substitution for margarine. We don't have any, and we use it anyway. And now the pancake or the pound cake doesn't rise as it should. And so it's clearly kind of unappealing. The flavor's not very good. And so that one was really where it was a harmful change in the recipe. I am going to, I can leave this last part to be kind of if you guys had any questions for me, if you wanted me to go over anything else. I wanted to just give you a little taste of stuff, and happy to answer questions. And I'll leave it on this one.